Morning children, and welcome back to biology class. Today we'll be discussing on transcription. Clear? What is transcription then? Yeah, it is the process of copying genetic information from one strand of the DNA into RNA. Here adenine base pairs with uracil instead of thymine. Okay? The DNA-dependent polymerase, RNA polymerase, it will catalyze the polymerization of RNA only in the 5'3' direction. And you also know that only a small part of the DNA is used in transcription, right? And the 3'5', the strand with 3'5' polarity acts as template. Okay, and 5'3' act as the coding strand. Okay, children, why both the strands are not copied into our RNA? What will happen if both the strands are copied? If both the strands are copied into the RNA, they may be complementary to each other, right? They may be complementary to each other and they may form a double-stranded RNA. And there is no process of translation into proteins then. Okay, right? Also, both the strands will code for different kinds of proteins if both the strand is copied into RNA. And it will complicate the process of, again, translation. Clear, children? So, you know, I told only a small part of the DNA is getting copied into RNA. And that small part, that structure is called as the transcriptional unit. Okay. Now, it consists of 3 regions. What are they? A promoter region, which is located towards the 3' end, and a terminator region, which is located at the 5' end of the template strand. Okay, and the structural gene is in between these 2 areas. Okay, right? So what are the 3 areas of the structural gene? 3 portions: a promoter gene, a promoter area, terminator area, and the structural gene in between them. Clear? Children, the promoter region is the place or the site where the RNA polymerase binds. The RNA polymerase is the enzyme, right, which helps in the polymerization of RNA. So, promoter region is the place where the RNA polymerase binds and starts initiation of the process, right? And the terminator is the region where or when the process of RNA synthesis stops. Clear, children? Children, you know, gene is called as the functional unit of inheritance. Okay, but you should also know that it is also the segment of DNA which codes for a protein or which codes for mRNA or RNA. Isn't it? RNA codes for protein anyway, right? So what is a gene, children? It is the segment of DNA which will code for RNA or protein, whatever you can say, right? Or it is, the gene is also called as cistron. What is that? Cistron. So what is a cistron or a gene? It is a segment of DNA coding for a polypeptide, is nothing but a protein, right? Okay, during protein synthesis. Clear, children? Now you should know that in our case, that is in eukaryotes, our cistron is called as monocistronic, whereas in prokaryotes it is called as polycistronic. What do you mean by monocistron and polycistron? You know the structural gene, a small segment of this DNA which codes for RNA, right? Which codes for only one protein, which codes for one small segment of RNA, which codes for single protein. Okay, whereas in prokaryotes, you know, there are many genes on the DNA which codes for different proteins. They have mostly circular DNA, right? Circular DNA. And there are many genes located here, many genes. So we call it as polycystron, right? And in our case, we call it as monocystronic. Clear, children? Okay. And one other difference between eukaryotes and prokaryotes are that in our case, in our monocystronic structural gene, we have split gene arrangement. What is that? We have split genes. What is a split genes? Here you can see there are introns and exons like that arranged in sequence. What are these introns and exons? Exons are the non-coding sequence. Exons are the coding sequence which will appear in the next mRNA, the mRNA which is transcribed, right? And introns are the ones which are non-coding sequence, Exons are coding sequence and introns are non-coding sequence. Children, the name itself is, you can just think opposite, right? Okay, so after in the form, when mRNA is formed or RNA is formed, the exons will appear in sequence and the introns will be all removed, should be removed. Okay, whereas in polycystronic structural gene, the genes are arranged in an adjacent manner. There is no split gene arrangement, children. Next we'll see transcription in prokaryotes. Children, transcription in prokaryotes and eukaryotes is an often repeated question, very important question, right? Okay. Now in bacteria, in prokaryotes, one more difference is there between eukaryotes and prokaryotes, children, which is in prokaryotes there is only one type of RNA polymerase in the process of transcription. Okay, and there are mainly 3 types of RNAs which are important for the next process, which is translation, which is protein synthesis again. Clear? Which are the 3 RNAs? Which is the mRNA, tRNA and rRNA. Clear? Which are they? mRNA, which is by transcription mRNA is formed, and transfer RNA, tRNA and rRNA, which is ribosomal RNA. Right, children? Now all the 3 RNAs are transcribed by the same RNA polymerase. Whereas in eukaryotes, we have 3 different RNA polymerase for synthesizing each of these 3 RNA polymerases. Okay, now we'll go into the transcription in the case of prokaryotes. Now the process usually contains 3 steps. It can be described in 3 steps, which is the first step is initiation, second is elongation, and the third is termination. Okay. Now these are sigma factors, rho factor, all these are actually proteins, children. Okay, so this sigma factor which will bind to the RNA polymerase at the promoter end. Okay, when it just binds, the initiation has started. Okay, now it just starts synthesizing mRNA. Okay, only what is the difference between RNA and DNA? The only difference is that instead of thymine, there will be uracil in place of thymine. That's the only difference, right? Okay, so what is elongation now? Elongation is the synthesis of RNA. These factors, sigma factor, just moves along the DNA and synthesize the RNA. Okay, and when termination comes, or when it reaches the terminator region, when the polymerase reaches the terminator region, there is another factor which binds to it, which is the Rho factor or termination factor, and the process of RNA synthesis stops. Okay, right. So you can just, in 3 steps, you can explain initiation, elongation, and termination. Clear? Children, in bacteria, transcription and translation can be coupled. Coupled means can happen together. It happens together because in prokaryotes there is no nuclear membrane, right? There is no demarcation between the cytoplasm and the nuclear membrane or the nucleus as such. So they have only a nucleoid in where you can find the genetic material. Okay, the DNA is used for transcription, and then by the time the transcription is completed, the translation can begin on one side. Clear? That's why it is said that transcription and translation can be coupled in the case of prokaryotes or bacteria. Whereas in eukaryotes, the transcription happens inside the, where does transcription occur? On the DNA. DNA acts as the template, right? So it happens in the nucleus. After which the mRNA should be transported to the cytoplasm, where the next process, which is translation, happen. Clear, children? Where does translation actually happen? Protein synthesis happen? Yes, you said it, which is the ribosomes, right? Ribosome is the place where protein synthesis happen, which is there in the cytoplasm. So the mRNA should be transported to the cytoplasm. Clear? Children, I told you there are 2 differences between eukaryotes and prokaryotes transcription. What are they? What were they? Yes, that is, in prokaryotes there is only one RNA polymerase which synthesizes all the 3 types of RNAs. Whereas in eukaryotes there is 3 types of RNA polymerase to synthesize each of these 3 different RNAs. Okay, now what was the second difference we told? That is, in prokaryotes there is no split gene arrangement. It is polycistronic, and also it is not split, which is there is no introns and exons, right? Whereas in eukaryotes we have a split gene arrangement, which means the coding and non-coding sequences are adjoined with each other, and the non-coding sequence has to be removed. Okay, and the coding sequences has to be arranged in sequence. Clear? Children, so RNA polymerase 1 is the one which transcribes rRNAs. What is rRNA? It is the ribosomal RNA. So which is the one which transcribes ribosomal RNA, children? It is RNA polymerase 1. Right. RNA polymerase 2 transcribes our mRNA. The mRNA, which is transcribed from the DNA. Children, I also told you that in last class that the mRNA formed is not perfect, right? It has to undergo some additions and all those to become a processed mRNA, right? Okay, and the mRNA formed or RNA formed from the DNA is called as hnRNA. What is that? It is the hnRNA which is formed from the DNA. Actually, it is the one formed. What is hnRNA, children? It is heterogeneous nuclear RNA, which has to undergo 3 processes, okay, to become into an mRNA. Okay, now after transcription, what's the next process? It is translation, right? And there are 3 types of RNAs needed for translation. Now, which are these 3 RNAs? They are the mRNA. mRNA is the one which is getting transcribed from the DNA. But in the case of prokaryotes, eukaryotes, the mRNA formed will be or is called as hnRNA. Okay, it has to undergo some kind of processing to become into an mRNA. Clear? Okay, and what are the other RNAs needed for translation? rRNA, ribosomal RNA, and tRNA, which is transfer RNA. Clear, children? And polymerase 3 transcribes tRNA. So, which are the ones? Say again. Polymerase 1, RNA polymerase 1 transcribes rRNA. Polymerase 2 will transcribe mRNA. RNA polymerase 3 transcribes tRNA. Clear? Okay. Now, what are the 3 processes which has to be undergone by hnRNA to become into mRNA? It is splicing. What is splicing? Splicing is nothing but the exons all are brought in together and introns all are removed by the process of splicing. Introns all are removed and all the exons are connected together to form a sequence or coding sequence. That is splicing. Second thing is capping. Capping, which is the cap end or head 5' end of the hnRNA, right? And here what happens? An unusual nucleotide called as methyl guanosine triphosphate. What is that, children? Methyl guanosine triphosphate. Guanosine triphosphate, right? So it is a nucleoside, right? It is added at the 5' end of hnRNA. Okay, and tail end. Okay, at the tailing means at what end? 3' end. At the 3' end, some nearly 200 to 300 adenylate residues are added at the 3' end. Okay, now when all these 3 processes are complete, now the hnRNA becomes or get processed or transformed into mRNA, messenger RNA, because now it contains the message. What is the message encoded inside this mRNA? What is the kind of protein to be formed? Clear, children? So what are the 3 steps which are formed, which are done to the hnRNA? Yes, splicing, tailing, and capping. Clear? Okay. So that is for today's class, children. Thank you, all of you.